Hi, welcome. Thank you so much for inviting us to speak today and talk about our research. Find the button. There we go. So my name is Miriam. I'm the research coordinator for the Maldives Underwater Initiative based at Big Census Laamu. And today I'm super excited to present to you our first results from our pilot project on monitoring coral reproductive cycles in Laamu Atoll. And my research colleagues on this project were Pitt, Ali, and the whole MUI team, and also Matt and Jamie from the Horniman Museum and Gardens in London. Just to give you a quick overview, we obviously all know how incredibly important coral reefs are, and we also know that they're under ever-increasing threat. So for coral reefs to naturally recover, we rely on the successful spawning, the fertilization, and then the settlement, and obviously the survival of the recruitment of the recruits. So for that, the knowledge of spawning periods and intensity is incredibly important for assessing the reproduction, the health, and the resilience of our coral reefs. And there's been a general perception that at the moment there's two major spawning periods in the Maldives, which is March and November. There's actually large data gaps in the spawning patterns in equatorial reef systems such as the Maldives. So this is our objective. We wanted to investigate the reproductive patterns of tagged and monitored colonies on the house reefs at Big Census Laamu to develop a baseline understanding of coral reproductive cycles in Laamu Atoll. You can see where we're situated in the Maldives. We're the southernmost atoll of the central atolls, and then our Oliveli Island is just situated between the largest channel of Laamu Atoll. Our survey site was our house reef. The house reef extends over 1.5 kilometers. But actually we focused on only tagged colonies, which I'll go into more detail in a little bit. Our house reef has actually remarkably bounced back from coral bleaching since 2016. So we now are at a hard coral cover of more than 40%, which is incredible in just those recent years. And we've identified around 37 different genera on the house reef alone. So that's quite a diverse reef. So the corals that we were tagging are situated in the red circles you can see here, but a normal survey dive would cover around 100 meters, and you can see the stretch of the normal survey dive over here. We tagged a total of 11 aquapora species over time. We focused on aquapora because they were heavily destroyed through the bleaching events, and also they're obviously most used in restoration projects, but also they're quite simple to sample for us as well. So we didn't want to only just look into the reproductive cycles on a species level, but also on an individual colony level. So every month, 3 days prior to the full moon, we would go out and survey for gamete development in the coral. So we have 24 different colonies tagged on the house reef that we take 2 small fragments of the colony and look for gametes. If we find gametes, then we obviously take that data on. We put the samples of each colony back onto the coral for it to allow to regrow back onto the colony. And the colonies with pigmented gametes, we then equip with gamete traps that then allows us to actually track any sort of spawning events on to a daily level if we're not able to go into the water. So we obviously then look into the literature and we estimate a spawning day and spawning time for each of the species that have pigmented eggs. And then we target these days around the full moon and we go diving at nighttime. And then when we go in the water, we collect data on the days after full moon, the hours after sunset, the setting start time, the duration of the spawning event, and also the number of individual colonies that spawn. And also we obviously collect data on any other colonies, on any other species that we don't tag. And that allowed us actually, so we started this project in February last year, and so far we have conducted 72 underwater surveys, which resulted in almost 100 hours spent underwater surveying, which is quite a big sample size now. Here I just want to show you a little overview of the variation of gamete pigmentation that we detected for different species. So you can see that in D. varicata, we've actually had quite pale gametes, so white and yellow gametes, the month of the spawning event, so only days before the actual spawning event. They didn't actually have any darker coloration, while for most other species, we actually detected this pink and dark brownish pigmentation for the corals. And you can see that in Aquapora humilis, you have pink pigmentation the month before the spawning event already, while in Laamu, this pigmentation actually only comes the month of the spawning event. And we were actually really happy that we detected gametes in 83% of all of our tagged colonies, which is really exciting. So quite the reproductive reef. We had 18 colonies of our 24 colonies spawning from 9 different Acropora species. And this is basically the overview that you get if you look at all of our tagged colonies. If you look at the red cells, that's all the spawning events that we monitored in water. If you look at the blue cells, that is basically suggested spawning times that we got from the information of the presence and absence of gametes from one month to the next. And there's actually really quite interesting data in here as well. You can obviously see some colonies from the same species develop eggs much closer towards the spawning event than other colonies. If you look at Acropora lorites and paniculata, some of the colonies only had eggs for 2 months before the spawning event, while other ones had them for 7 months. So it's quite interesting. And there's also some temporal reproductive isolation going on, also in paniculata and lorites. So different colonies from the same species spawn at different times of the year, so over a few different months. So from that data, it allowed us actually to identify that Acropora humilis takes about 9 months from one spawning event to the next, and 10 months for Acropora digitipara. And then we had the majority of spawning events happening at 6 days after full moon and 7 days after full moon, and only one colony each spawned at 3 and 5 days after full moon. And I mentioned earlier that we also collect data on colonies that we haven't tagged. So all of that data you see on the top part of the table, and then still our data from the tagged colonies you can see on the bottom part. And there's a clear pattern visible. So again, we started the project in February last year, and you can see we had immediately spawning events happen in March and April, and then we had a gap from May to September. And then from then onwards, it was basically spawning every single month. So we had some busy times, some late nights. So yeah, spawning in October, November, December, January, February, March, April. So not just November. So that was really exciting for us. And I don't really have time to go into synchrony spawning event discussion, but we had a mass spawning event in January where we had multiple species of 9 different Acropora species spawn at the same survey dive, which was really incredible and quite different to all the other spawning events that we had over time. And then obviously we know that environmental factors play a major role in affecting the timing of the spawning event. We have temperature loggers at the house reef at 5 and 10 meters, right next to the tagged colonies, and we know that temperature is obviously a really important factor. However, for the spawning events of our tagged colonies, we actually couldn't detect any clear trend. We also didn't have a very clear trend of the temperature on the house reef over time. So this is 2021 data. So just to conclude, we believe that we provided the first insights into reproductive patterns of coral species in Laamu Atoll, and there's definitely not just the 2 spawning events in March and November. And we also found that gamete tracking is a very easy and time-efficient way of getting simple spawning data, simple to collect and simple to analyze on a monthly basis. But if you want more in-depth knowledge on the spawning events, then you would need to go in the water and collect that data in the field. But yeah, basically with this data set, we now have a basic framework that we can then use for further data-driven studies to understand the reproduction, but also the synchrony, and investigate the external factors that affect the spawning events as well. Thank you so much.